All right, in slide 10 here, we've got the wrap up of the Griffiths experiment. And basically what I want you to write down for this uh, is that Griffiths experiment shows a transference of the dead S-type bacteria that that transforms the live R-type bacteria into basically virulent S bacteria. Virulent meaning disease-causing bacteria again. All right. And the next slide is all about the Hershey chase. Okay, so if I go back up here to the timeline, you can see that Hershey chase experiment in 1952 uses the radioactive labeling to prove that DNA is responsible for heredity. And how did they do this? And again, this is kind of mind-blowing to think about even now. I mean, this was back in 51, 52 that they decided to do this experiment. And they weren't really scientists that were out to prove beyond a shadow of a doubt that genes were in DNA. They were out to prove where were genes located, period. If it happened to be in DNA, great. If it happened to be in the proteins in the membrane, fine. But they wanted to put to bed at the very last little bit here where genes were located. And so now if I get down to the slide here, so here you need to put Hershey and Chase's experiment. And let me go over what their experiment entails. All right, their experiment starts with a phage, which is a little mosquito-like organism here. It's a virus, but just like a mosquito will land on the surface of its host, bite its host, so does a little virus. It lands on, in this case, a little bacteria, which is a phage. It's what we call a phage, is a virus that affects a bacteria. And in this case, we know that it puts the DNA that it has into the little bacteria. That DNA will then tell that bacteria, hey, you're doing a great job what you're doing, but I want you to stop everything. Stop building this, stop building that, stop building this, but use all your building materials that you currently have, and I want you to make more of me. This is how viruses reproduce. They don't reproduce by asexual means. They don't reproduce by sexual means. They reproduce by invading a host, having their DNA tell the host DNA, stop what you're doing, make more of me. And that's exactly what happened here. Okay? But how did they prove it? Well, they proved it by extracting the DNA in the virus. And if I go back up here to this slide, and I don't know that I can get in here. Yep. All right. So there we go. If you can see here, I've got a sugar binding to a phosphate, binding to a sugar, binding to a phosphate. This is the DNA backbone. So it's the backbone is consisting of phosphates and sugars. And what they did was remove these phosphates and substitute in a radioactive phosphate, which was, I think, ingenious. To think that, okay, yeah, I'm just going to go into a microscopic organism to find its DNA, extract and just take out its phosphates, and I'm going to substitute new phosphates in there. I mean, to me, that's kind of mind-blowing to think that, yeah, you could do that. And even nowadays, with our technology, it's still kind of baffling that they are able to do that. And this was back in 51, 52. So they perfected how they do that. Then they would go into the virus's DNA, change out its phosphate for phosphate 32, which is radioactive. And they said, all right, that will solve our problems. But then they got to thinking, wait a minute, that's going to give us the same situation that Griffith had. So we still wouldn't be able to disprove that is the proteins in the membrane that is not carrying the genes. So what they did is they found a molecule that is radioactive that could bond to the proteins that are in the cell membrane, and that is sulfur 35. And so they got the proteins to bond with this S35. And now they hypothesized, all right, if we have this virus that now has S35 on the proteins that's in the cell membrane, and we have this DNA that is radioactive with P32, and we have it now infect the bacteria, and the bacteria now makes new little cells. It's got to use some of the P32 and it's got to use some of the S35 to make new little organisms. If the new little organisms had P32 in them but no S35, this would basically say, all right, the transference of genes is in the DNA going from the parent virus to the new viruses. If the majority of the genes is in the S30 or is in the proteins that's in the cell membrane, then the majority of the S35 would be found in the new little viruses. And what they found after allowing these guys to infect the host and have the host reproduce them was very little of the S35, if any, was found on the new little viruses. That their little bodies were all made up of stuff that was in the bacteria itself and not radioactive stuff. And most, if not all of them, had some traces of the P32, which is saying that this parent had to pass its genes down to this offspring. And the way it did it was through its genes. And the genes then were proved to be in the DNA of the molecule. So the comment I want you to make on this is the Hershey-Chase experiment proves the transference of genes through DNA. Okay, so that's the transference of genes is through DNA. And that was proven by the Hershey-Chase experiment. This kind of puts the lid on the coffin. The part that nails the lid down on the coffin was when Watson and Crick a year later found the structure of DNA and was able to put that structure then to use in describing how genes are formed from the variations of the bases. So all of the A's, T's, C's, G's, and their different arrangements will form a code for the gene, which will then code for a protein. All right, and the last slide here, slide number 12, polynucleotide chains have nitrogenous bases linked to a sugar phosphate backbone. So what I'd like you guys to put down for a comment on this is poly means many, nucleotide is nothing more than a phosphate, sugar, and a base. So this is many nucleotides that are bonded together will form this polynucleotide. Okay. So again, a nucleotide is a phosphate, sugar, and a base. Poly means many. So in this case, you have many nucleotides that are all bonded together. And that forms the backbone of the DNA. All right. And that will wrap it up for this video, and talk to you later.